Welcome to the Norovirus chapter of the Bad Bugs of Produce-Associated Outbreaks series. In this lesson, you will learn about the basic biology and ecology of norovirus. Specifically, we will cover the sources of contamination and biological traits of the organism that will influence your investigation plan. By the end of this module, you will be able to identify basic biological characteristics of norovirus associated with produce outbreaks, identify common sources of norovirus contamination of produce, list the primary reservoir for norovirus, understand the human health impacts of norovirus. Throughout this presentation, you will see this magnifying glass icon. This indicates an investigator tip that will help you as you plan and carry out your investigation. Norovirus is the most common cause of foodborne disease in the U.S. Data from the Centers for Disease Control and Prevention indicate that norovirus causes an estimated 21 million illnesses and contributes to 70,000 hospitalizations and 800 deaths annually. In general, viruses such as norovirus, hepatitis A, and others can only produce in living cells. Viruses can infect all types of organisms, including plants, animals, which includes humans, and bacteria. They are found in every ecosystem on Earth. Here's a tip for the investigator to keep in mind when dealing with a norovirus investigation. These pathogens may be resistant to unfavorable environmental conditions, such as heat or chemical disinfectants, and therefore may survive in the environment or on surfaces. Norovirus is very environmentally hardy. For example, norovirus can persist on human hands for at least 2 hours, on surfaces for 3 to 6 weeks, in water at room temperature, and in the dark for 61 days. It can persist even longer on surfaces at cooler temperatures. Here's a tip for the investigator to keep in mind. Norovirus can survive relatively high levels of chlorine, varying temperatures, and can survive on practically any surface. All of this will influence where you will want to collect samples. A very important thing to recognize is that humans are the only known reservoir for norovirus. This pathogen is extremely contagious, requiring as few as 18 viral particles to initiate an infection. Transmission route for norovirus is fecal-oral, meaning that it is passed from one person to the next through fecal contamination of water, soil, food, or surfaces that contact food or raw produce, whereby the virus enters the new person through the mouth. Here's a tip for the investigator to keep in mind. Infected people can shed virus for up to 4 weeks after being sick, so be sure to ask about worker health prior to the outbreak. Produce can become contaminated with norovirus through contact with contaminated soil, irrigation water, hands, or equipment. Here's another tip for the investigator. When looking for norovirus, you will want to focus your investigation on those places where human fecal contamination is a possibility, including worker health and hygiene practices, water sources and use of or contamination with sewage, hint, think of soil and soil amendments, and equipment that people touch, such as gloves or knives. This chapter has provided an overview of the produce-associated pathogen norovirus. Norovirus is a human pathogen. The source of norovirus infections is human fecal or vomit contamination. Norovirus is usually spread through poor health and hygiene practices or through contaminated water. Thank you for watching.